Okay, so thank you very much for accepting our invitation. I actually become familiar with your company during the sleep meeting. Kun and I visited the Neurosum, I guess, at that time, and we saw
That's
that
right.
headband.
That's right.
Yeah, that headband which has also the stimulation. And we noticed that it is very close to what we are doing because we are also, so a general overview about our center, the focus is on sleep and also dementia. We are doing lots of works with EEG signals. So, for example, one thing that I saw your device also has is like real-time sleep staging. We also published a paper last year for real-time sleep staging using single-channel EEG. And we are currently working with a big cohort in South America, in Latino countries, people with dementia. And we are working on developing EEG biomarkers for them. And the next step for us is to also do some kind of stimulation to like improve their sleep quality and ideally improve their cognitive performance. Right now, one of the funds that we have is for developing biomarkers for predicting future risk of dementia, not detecting people who already have dementia, but people who are normal, but we predict if they get dementia in the next 5 years or not, based on their sleep time, low intensity EEG. So I think there are lots of similar interests here. I don't know if Kun, you are able to introduce yourself and other members are now able to introduce, or should we keep it for the end if you are not still at the office?
Well, I would say let me introduce our company.
Yeah, sure.
Rather than each person, because we have too many probably online for that. So
yeah, that sounds good.
Yeah, good. So my goal for today is to talk about synchronizing slow oscillations, the big slow waves of deep sleep, with the goal of extending deep sleep and really enhancing the depth and duration of stage N3, the deep stage of sleep. And we are the Brain Electrophysiology Laboratory, and we've created a new company called Neurosum, a corporation to build medical devices specifically for sleep assessment and therapy. And so we have a strong interest in aging and dementia as well. So I think, as you mentioned, there's quite a bit of overlap between our efforts and interests and the work in your lab. And I think that should become clear as we go through this. So the Brain Electrophysiology Lab was first established in the University of Oregon many years ago. We became EGI, focusing on high density or dense array electroencephalography. And we retained that focus. We sold EGI to Philips a number of years ago, and then established Bell really for advanced software development. But because Philips kind of failed to carry forward with EGI, we've now developed a complete hardware system. And so you'll see that in the course of our science work that I'm going to emphasize here. And I have kind of an ambitious set of goals that I've put into the slide for looking at how to synchronize the slow oscillations of deep sleep. And I won't go into all the detail now, just so we can get through the talk in a reasonable time. But the main points are, we want to understand the slow oscillations neurophysiologically. And there, the high density helps us to understand the sources of the slow oscillation of deep sleep, which we do think are critical for normal memory function. So that when the slow oscillations decline with aging, then there is a corresponding decline of memory. And of course, eventually it becomes catastrophic in dementia. But as we're going to show, The decline of deep sleep and the loss of memory are normal neurodevelopmental processes of aging. They may not be attractive or beneficial, but they are actually developmentally predictable. But our goal is to reverse the decline of slow oscillations and to improve the synchrony of slow oscillations with applied external electrical current. So I'll show that effort and some of our recent success in doing that. And let me just emphasize that please break in at any time. I really haven't practiced this talk, so it's going to be informal and easily interrupted if I'm not clear about something or you want to go more in depth. And I've tried to structure it for a half an hour so that we would have time for discussions and interruptions. Here's our current high-density EEG system, the Bell System 1. It's a 280-channel advanced geodesic head web. And a number of the advances we've made allow us to do recordings all night in sleep, including at home. And part of the goal of making this portable and easy to use was to make the high-density amplifier battery operated and wireless, so it could link to the home internet and provide two-way communication with our cloud server. The question really for high-density EEG is source localization. How do we understand brain activity, not just EEG signals at the head surface, but the sources, the generators, which we think are almost certainly cortical sources. So we want to map the electrical fields of the cortex, and the basic physics are that those electrical fields are perpendicular to the cortical surface. And because the cortex is wrinkled, we have to understand those wrinkles and actually quantify them exactly so that the dipoles that we're localizing, the dipolar fields, must be perpendicular to the cortical surface. And you can see, given the individual wrinkles that we have, the best localization will be with an individual head model where we can characterize those wrinkles. And the perpendicular electrical fields of the cortex with a high density electrical source localization. So we've worked on this problem for many years, and just briefly, our main approach has been to improve the anatomy that we're localizing to. And this includes the cortical surface, as I've tried to emphasize, but also the conductivity of head tissues, where the skull is a particular problem. So we want to know the dipoles, and you can see the region of dipole placement. All of the dipoles, for example, we may have 2400, 4800, or now 9600 dipoles that we're attempting to compute from the inverse of the 280 channel data. But their orientations need to be considered, and the intervening tissues that impede the electrical current getting to the surface electrodes need to be understood. And so the cortical surface has to be extracted, and we have to characterize each of the tissues, including the skull. So if we don't have a CT, and usually we do not because it involves ionizing radiation, we fit an Atlas CT that's close to that person's head shape, and then we warp it to their MRI, specifically their T1 MRI, so that our computational model of the electrical inverse includes all of the conductivity of the tissues. So we integrate those within our software package called Sorcerer. So Sorcerer is really the best anatomically specific EEG source localization package because we've accounted for all of these factors. So the other fact was to give good visualization of the EEG on the cortical surface, but also to have a user interface so that each user could be trained on these basic physics of how cortical sources generate electrical field, which is seen in the EEG. And finally, we've wanted to integrate this within laboratory operations that could include high performance computing, database archiving of the EEG, and individual analytic routines where each lab could build its own unique processing workflows. And we call this the forward-looking operations workflow or flow. So let me illustrate high-density EEG and the perspective it can give on underlying features. And I'm going to focus on the large, slow oscillations of sleep, often called slow waves. And either oscillation or slow waves kind of misrepresent these phenomena, these large, slow oscillations, because they're not oscillations in the sense of sine waves. They can look like that to kind of a low density EEG, not studied very carefully. But actually, it turns out that each large slow oscillation is a transient. It's not really a sinusoidal feature. And you can see it in the denser, high density EEG over here. The brown line in the EEG shows a single punctate event, a slow oscillation. Actually, you could also call this a K complex because they share a lot of features. The main feature is a big negativity that's typically toward the front of the head, not always. And it's followed by a smaller positivity, a second waveform that looks to be kind of a reversal of the first big negativity. Now, one of the things, if you don't understand source localization, you might describe this as a frontal negativity, and you might ignore the positivity in the back of the head. And in fact, a number of leading labs have done just that. And so they saw the big negative field moving over the head as the source actually changes and described it as a traveling wave that was in the frontal lobe. And this actually turns out not to be the case for reasons you can see if you look at the head field distribution on the right. Exactly coincident with the frontal negativity is a posterior positivity. That suggests the source is dipolar. And in fact, if you look at the inversion between the negativity and the positivity, you see this is in the midline, middle regions, such as in the temporal lobe. And in fact, that is the source. That is the generator of this field, not the negativity or the positivity. Now that comes out naturally when you conduct an electrical source analysis, an inversion to have a putative cortical source for creating the surface fields. So that's what we do in Sorcerer. We do this source inversion. And on the left, you can see the big negativity. So this is 40 channels taken from the front of the head. So everything looks negative. But if you plot the head field distribution, as in B here, you see that there's a frontal negativity, but a posterior positivity exactly coincident in time, suggesting that the source is somewhere in the middle of the head, and particularly around the temporal lobe, the anterior to medial temporal lobe. And this is what our source localization with the Bayesian multiple sparse priors, the best algorithm that we found for source inversion shows. So in C and E, you can see that the source that explains this head field distribution is actually in the right medial temporal lobe. And this is a pretty tight focal source in the parahippocampal gyrus. And it's not untypical of the people that we look at. So the main point is that slow oscillations do look very large and impressive, but they're not diffuse across the head. We think there are up waves and down waves, up states and down states that are local in the neocortex during the slow oscillation. And there's plenty of intracranial recording that suggests that. But those are fairly small field patterns that are difficult to localize in the EEG. The major EEG events we see are in limbic regions. Here's another person. Again, if you look at A, you're seeing the voltage field patterns in the frontal areas that are negative going. We have the typical frontal negative, posterior positive pattern, suggesting an anterior temporal source. And then here, if we do the source localization, there is activity in the anterior temporal, but also the caudal orbital frontal. Here, area 25. That's, again, a typical generator. The other feature that's interesting here is that when the wave inverts, when the frontal negativity kind of repolarizes or whatever it does, there's a field inversion and a frontal positivity follows. And if we localize that, it shifts from a source in the caudal orbital frontal to a sink. So there is a reversal in the following positive wave. But by and large, in the study that we did, this is Morgan et al. In Sleep Research 2021, very typical. The generators of slow oscillations are in limbic areas. And we can make a flat map to characterize this. So for example, if we look at the count, the number of slow oscillations localized to each brain region, and the top image shows the Brodmann areas of the cortex, the bottom area shows the density or the number of counts of slow oscillations over this sample of 20 individuals that we localized. The density is strongly in limbic regions for the slow oscillations of deep sleep. Now, part of our interest is to understand why. What does it mean that there are limbic generators? And how do we understand this neurophysiologically, but also functionally, to the functions of slow oscillations and deep sleep in memory consolidation? Because the evidence is quite good that normal consolidation of memory requires the non-REM sleep, and particularly the stage 2 and stage 3 features of spindles and slow oscillations in order to consolidate explicit memory. We're very interested in the neurophysiology that suggests that slow oscillations are synchronized and coordinated by the claustrum. And this might help to explain why we see the strong ventral limbic control, the amygdala and orbital frontal cortex, as being the primary control systems for the slow oscillation of deep sleep, because the amygdala claustral complex seems to be involved in the widespread synchronization. And with widespread projections from the claustrum to limbic cortex as well as neocortex, this might be a way of understanding the neurophysiology. And then the function seems to be important, excuse me, not only to normal memory consolidation, but also the decline of memory that occurs with the loss of deep sleep in aging, and particularly in Alzheimer's. Excuse me. So I'm pointing now to the classic neuropathology of Braak and Braak, showing that as Alzheimer's progresses, the neurofibrillary tangles and plaques accumulate in limbic regions. And this really clearly appears to relate to the memory impairment that happens in Alzheimer's disease. So the link between slow wave decline in aging and dementia seems to point to these limbic regulations of memory consolidation in the normal brain, so that when slow wave sleep particularly declines in aging, there is a loss of the memory consolidation and therefore memory impairment. This is a recent study I'm pointing to from the Framingham Heart Study that looked at normal senior citizens, normal old people without MCI with over time. And what they found, which again is a normal progression, the ones that lost more of it were at risk for dementia. And in fact, in this remarkable ratio, that a single percentage decline in a year predicted 27% increase in the risk of dementia over the subsequent years at that point. So we're very interested in understanding the nature of these mechanisms and how to appreciate them. Here I've plotted the amplitude of the slow wave sleep, and you can see the normal developmental course involves a decline in adolescence and a continuing decline in aging. This is from our own machine learning studies that we applied to the Cleveland Family Studies database available at NCRR. And you see the decline in adolescence is quite remarkable. It's a normal neurodevelopmental process to lose deep sleep. But then about age 30, there's a further decline for many people, but not all. Some people maintain their deep sleep and aging. Many people do not. And we think the evidence points to the ones that lose it are the ones who are on the road for cognitive decline in aging. The question is, how could we improve this? This became really the goal for Neurosom and creating a medical device company that could use a simple headband to synchronize the slow oscillations of sleep. What we did was to reverse our computational modeling. So now we're not trying to localize the sources of the slow oscillations. We're trying to inject current, to apply current into the head in an optimal pattern for targeting these deep limbic regions that seem to be the sources. And it turns out that when we do this computational modeling, what we find is that we don't need the high density array to have a pretty reasonable targeting of the deep sources. And in fact, if we choose the optimal electrodes, we can achieve a fairly good targeting of the limbic generators, anterior temporal and caudal orbital frontal particularly, with just a few electrodes. And that's what this simulation shows. On the left is a simulation of the original TES synchronization of slow wave sleep by Lisa Marshall in her group, using fairly high current levels of like a milliamp. And that's enough that it's a little bit noxious. What we showed on the right is we can have a fairly good approximation with half of that current. So the question was, if we cut the current and only use half of a milliamp or 500 microamps, could we still synchronize the slow oscillations of deep sleep? And that's this study, Hathaway et al. We targeted the sleep stages, and here it was before we had our machine learning that was working adequately. So what we did was have a research assistant looking for N2, and as you know, you can see the spindles and K complexes of N2 are pretty easy to detect by visual inspection. After 4 minutes of N2, we judged that the person would be typically ready to go into N3. We turned on the stimulation, again manually. And what we found was an increase of N3 that was small but highly significant. In this study, it was 17% increase in N3 on average. And interestingly enough, it not only increased during the stimulation, but it was stronger than those in the sham group throughout the night, the other cycles of N3. So the indication was that it was fairly reasonably easy to synchronize the slow oscillations. And our goal is to understand the pathophysiology of the N3 loss and what it means for aging. Now, here I'm showing an example of the clearance of toxins, such as amyloid beta and tau, that is achieved by the brain's glymphatic system. And there's fairly interesting evidence that the brain's glymphatic system is strongly active during deep sleep, during N3 sleep. Some of the evidence actually came out of MGH, Laura Lewis's lab, where they focused fMRI actually measures to assess the CSF clearance by looking at the base of the brain and to estimate the clearance of CSF. And what they found, shown in figure E here, or patch E, is that during slow wave sleep, there are big fluctuations, and you might think of it as pumping CSF out of the brain, such that, for example, in figure G, there is a much higher increase of the CSF excretion from the brain or clearance from the brain in sleep than in waking. And this appears to be specific to the deep sleep. Our goal then was, how can we enhance the synchronization of slow oscillations, the electric sleep therapy, as we call it, at home? Because we know that sleep in the lab is somewhat unusual. It's not like sleep at home. So our goal would be to have a sleep therapy at home. Here's one of our first prototypes. It's not particularly elegant, as we wanted to bring the whole technology of a battery-operated method for synchronizing TES. That's not so hard to do. But we also wanted to integrate it with machine learning. And it turned out that's not so hard to do these days. We have worked with convolutional neural nets for a decade, and the hardware keeps getting cheaper. So NVIDIA has some remarkable chips that allow us to implement TensorFlow and other machine learning for our convolutional neural nets to detect the sleep staging in real time. And I think you guys are familiar with that from your own work. So we set up a nanocomputer at the bedside, and we interfaced it with our simple Bluetooth headband. And this became really our medical device delivery to build from our high density EEG to make a simple headband to do EEG classification, and then TES therapy for synchronizing deep sleep at home in an automated closed loop package. And that's really the framework for Neurosom, the company that we've now established to bring this to medical devices with FDA clearance for specific indications. And our first indication will be healthy adult sleep, because that's really who needs it. It's not after you have MCI or cognitive decline or dementia. It's kind of too late then. It's when you're still healthy, but getting old. That's when the deep sleep therapy is necessary. So that's really the framework for Neurosom. And I will mention that if you're interested in investing in Neurosom, I have a brief commercial here, that we are available for investments right now on the RedCrow platform. Here's some of the results of our first home sleep study. So our first goal is to say, okay, we think, an understanding of the slow oscillations of deep sleep and how to target them most effectively. Let's prove it. And let's start with healthy adults. So here we have a range of healthy adults. A lot of these at this stage of our kind of preliminary analysis are younger adults, so they're pretty easy to work with. But they're also equivalent to the Morgan et al. And Hathaway et al. Samples or earlier published work in they're fairly young. Not all university students and recent graduates, but a lot of them. A few older people that do have good N3 synchronization, but we need to improve the aging sample. What we find is that at home, people sleep much better, and the N3 enhancement is much better. So rather than a 17%, with this first interim analysis of the home sleep, we get a 44% increase in N3 enhancement. So it's not so hard. It works fairly well with healthy adults. We have funding now from NIA, National Institutes of Aging, to bring this to adults with mild cognitive impairment. And we're working with Ruth Benka at Wake Forest University and with Miranda Lim at Oregon Health Sciences University. In a phase 1 trial, we were able to train the seniors with MCI that we recruited from the Wake Forest, Winston-Salem, and Portland, Eugene community, with help from family members in many cases, to use the sleep wisp at home. So we showed we could get good data in a home environment with this technique. So we do want to bring it to aging as well as to bring it to healthy adults that we think is a major target. And the goal is to improve glymphatic clearance, to improve the washing out of the toxins. Amyloid beta and tau are kind of the major ones, and their downstream effects on neurodegeneration, which can be indicated by glial cell measures such as GFAP and the neuroinflammation that's part of the pathophysiology. And here we've teamed up with John Nailway and his colleagues at Sonus Biotech, also located in the research park at the University of Oregon, right next door to us, to measure markers of the glymphatic clearance. And we're particularly interested in doing frequent measures, even daily measures, with nose sponges and nose swabs, because the nasal secretions seem to be a very good route for routine daily assessment. So that's really the overview of our research program. We have basic science interest in understanding the mechanisms of slow oscillation, how they figure in memory consolidation, and how they can be maintained at healthy young adult levels as people are aging and moving on to the later years of their life. So I'll stop there and open up for questions at this point.
Thank you so much, Dr. Tucker. I mean, I apologize, I'm a little late. I think there's many fantastic aspects regarding the you know, slow wave sleep and how that linked to the dementia. But of course, I do have a lot of questions. I mean, maybe I can start with very general ones. Of course, well, for the people who focus on like sleep physiology, sometimes we ignore what really happened within each sleep stages. So you make some of the great points here. I think you, for example, you are not looking at the only within the sleep stages. You look at, for example, the amplitude of slow wave, you know, amplitude of slow wave form, right? I think this is something which we also want very interested, right? It's not just like you in that sleep stage, but what happened within that sleep stage. That's what exactly we are also very interested. I think regarding the pathology of dementia, I think that this is something also open question for the field. I mean, it is possible just because you do not have a good sleep. When we say good sleep means you do not have enough deep sleep stage, right? Or what may happen is within certain sleep stages, like what you're gonna to show you some of the biomarkers showing, okay, even though you're in that stage, it's really not your brain not function or some pathological condition affect the brain activity. So that is something very intriguing for me. So what is your current focus for that new trial you presented? Is that the focus on? Can you elaborate a little bit more about the what is the like milestone you want to achieve? Use that your new device and the stimulation. What is your milestone?
No, it's a very good question. And to your earlier points, we fully agree that we need to understand the neurophysiology of sleep. It won't be enough just to get more deep sleep, although that is an outcome that we're focusing on. Mm-hmm. We have to understand the change in mechanisms, and particularly for memory consolidation. And for example, and I think one of the most intriguing findings is the loss of coupling of slow oscillations with spindles that happens. That the healthy young brain has a very tight coupling of spindles with the slow oscillation waveform. And furthermore, from animal studies, we think the hippocampal sharp waves are nested and coupled within the spindles themselves. So the health of the brain is not just the stages of sleep. We fully agree. And so we're trying to look at spindle slow oscillation coupling as an insight to that. But in our first trial here, we're trying to be systematic and not too creative, because we're trying to get NIH funding. So being creative just doesn't work with NIH. So our goal is to improve the length of deep sleep, the amount of N3. That's the simple goal. But we're also assessing memory as on a regular basis. So we have a verbal learning paradigm similar to that used by Marshall and others in looking at the relation of deep sleep and memory. And so those are our outcome measures: the length of N3 and the capacity of memory measured over time. And we'll do it in a sham control. That's very important, so that nobody knows what the stimulation is, except once the data is analyzed.
Cool. Yeah. Oh, great. I think one thing for those MCI, your current patients with MCI, I think I may miss some of your presentation. Is that the stimulation target on the whole brain or target on some of the specific region, like what you presented, the source of the slow wave sleep, or is there any target?
Yes, that's a key element, is that we use the high density EEG to tell us the source of the slow oscillations. And so you can't just use TES with the brain generally. You have to target the source of the slow oscillations, and they're fairly focal features. They're localized to the limbic regions, and particularly ventral limbic regions, perihippocampal, although we think they're mostly synchronized by the amygdala and the claustrum. So orbital frontal cortex and anterior temporal lobes are specifically targeted by our headband electrodes.
That's cool. Yeah, that's exactly. I mean, I think limbic. I think that area you presented. So is that like a very established that even in the MCI? I know you showed the paper 1991. They show in the AD they have a fact that, but for the like recent study, is that established the limbic also system also the first region? I don't think so, right?
No, that's right. We know the pathophysiology is limbic in Alzheimer's disease. And we know the limbic regions are critical to memory consolidation from a lot of evidence, human and animal. But I think our data are the first to show slow oscillations generated in limbic regions. And it's not widely accepted that that's the pathophysiology. And in MCI, I don't know of any evidence that relates to that directly, except for ours.
Right. I mean, MSCI is a little bit tricky because many people have MSCI, but not necessarily any like what's the underlying neurophysiology changes, right? So that's actually my follow-up question. When you include the people, are you gonna do some of other, brain scan using the whatever the MRI or other things to select those people with some of the neuroanatomic changes certain of brain? So
No, in this trial, we will not do the MRI. I think that could be very interesting to relate the anatomy, mainly because if we could do enough high density EEG, we could localize their EEG changes, which are complex and not just the loss of N3 to the MRI features. That would be a very good study, but we don't have that in our capability now. What we are doing to understand this difference between MCI of more healthy aging, which is very typical, and the MCI of the progression to dementia, which is very unhealthy, that distinguishing factor is very important. We're trying to take biomarkers, and this is where our collaboration with Sonus Biotechnologies is going to be very important. We're measuring we're setting up these measures of nasal secretions, which we think are a good way, and we're trying to do it daily. Now, we're just getting this MCI trial started, and we're not funded for this, so we're trying to bootstrap the biomarkers on that project. We have a new project, ARPA-H, that would explicitly focus on healthy seniors and their biomarkers. But the biomarkers we're looking for will be amyloid beta, the 42/40 ratio, tau, phosphorylated tau, which we think is very pathognomic, and some of the neuroinflammation measures, GFAP, NFL, to give us a clue of the neuroinflammation that results. So if it works, then we'll be able to distinguish those people with MCI that are on a short course to dementia, compared to those that might have a neurophysiologic impairment of memory because their sleep is poor, but aren't quite so predisposed for the biomarkers that predict the onset of Alzheimer's disease.
True. Yeah. So of course, I want to shorten my. I have still lots of follow-up, but maybe I focus on the some things which kind of really want to apply to our study in Latino American countries. So well, first, I still want. I'm wondering. I know your study is still ongoing, so you trying to establish that those device stimulations can be used for the people with MCI, right? So
That's right.
Can you help us understand that so far any difficulty? What kind of difficulty you think will stop those potential application to the people with mild cognition, like we are focused on the Alzheimer, but also frontotemporal, dementia there. So do you see any possibility, or you think, oh, maybe that's too much?
I think
we cannot hear you, Dr. Tucker. Is it cut it?
Okay, now can you hear me?
Oh yeah,
Yes.
that's much better.
Good. All right. What's important is applying the headband. We try to make it as simple as possible, and we could improve it, I think. But right now, the WISP, the Wireless Interface Sensor Pod Headband, requires some training and skill to apply at home. We find some seniors with MCI do it well, others need help. So it's important to have good electrode contact. We have simple stick-on baby ECG electrodes, actually, and they're on skin. There's no hair, so it's fairly simple to apply the headband, get it on in a good place, and have reasonable impedances, which we need for applying current. So what we find is if the every patient in our group of MCI has somebody at home to help them. Typically, it's a spouse, but it could be a caregiver to help train that application of the head web or the headband.
I see.
And
I see.
then it's very important that we do a data quality check. So we routinely check the data quality as it's then uploaded from the dock, which is the bedside computer. So we have to run routine quality checks to be sure we're getting good data. But with those protocols, it's fairly easy to do data collection in the home environment. And what you saw from our data is people sleep way better at home, even if they have sleep problems. If you bring them into the lab, you don't get good data. I mean, lab sleep studies are just anomalous.
Yeah,
they're not like home sleep.
Exactly. I totally agree. We did a lot of in laboratory human study. It took like a few days sometimes to get used to a new environment, and depends on the person. I totally agree. So that's actually one of the things you remind me. For your like protocol or devices, maybe what I'm thinking is maybe you can focus more about those people with some of the sleep problem will be more better, right? To enhance your the effect size. I think I don't know if you in your population you only focus on those you know, generally without the sleep problem, or some of them with some of the more severe the sleep, for example, sleep apnea, other things, you know. Anyway, so that is a thought. But I agree with you. I agree with you.
That's a real question. So we right now, in fact, as we've set up our crowdfunding for Neurosom, our focus is to look at insomnia.
Yeah.
So we've met with the FDA and looked at the indication for insomnia and what would be required for a trial. And we're very interested in that. We would like to start a trial on insomnia. We need funding. We have not been able to get it from NIH so far. But that is the group we would like to look at. And we know they're going to be more challenging. So people with insomnia, it's not just that they're missing deep sleep, although their lack of deep sleep is very important to the anxiety that results from poor sleep. Poor sleep has a causal influence of increasing daytime anxiety, but there's a vicious circle that then they're anxious and can't fall asleep. So we think that insomnia is going to be difficult. It's going to take a coordinated effort to understand that. And we think that improving sleep will be more difficult in insomnia, particularly with comorbid anxiety, which is very frequent.
Yeah. Nice. Thank you so much. And also one more follow-up question regarding, I believe your patients is mostly white people, is correct?
We recruit from both Winston-Salem, North Carolina, which there'll be more Black people in that community, and Oregon, where we have a fairly reasonable population of Hispanics, but not so many Black people in Eugene. Actually, in Portland, Oregon, another recruitment site, we have a more heterogeneous population across different ethnic and racial categories.
I see, I see. Well, anyway, so in my mind, as I mentioned, we do have some ongoing study. Maybe in the future, I don't know. Your phase one, your study is that the NIA STTR or SIBR, right? SBIR, sorry.
SBIR, that's correct.
So how long your phase one? I know, I hope you can get phase one and go to phase two, of course. But do you think you still phase one will be last another year or two or something? Or what is the duration now?
So we've completed phase one, and we just started phase two funding in May. So we're now setting up phase two work, and that'll run for 2 years.
Got you. Okay. So yeah, we hope it is possible you are very successful, use your new device and then stimulation. And then hopefully, I think we can talk a little bit more. We can apply to people in Latin American countries, like, Latinos. And then that is exactly my question related. So do you think your system, for some other reason, can be a little bit different between, for example, white and black and then Latinos? Do you think any like because of the skull and other physiological difference? Do you think there'll be issue?
No.
No. Okay.
No.
Great.
So the headband system, it's actually not an issue for any of our measures. We have a lot of experience with different ethnic groups, even with the high density EEG. You can have a big afro, a black person with big hair, and it's not a problem. It takes a little bit of technique for applying it. Some hairstyles, such as cornrows, braids that are tight against the skull, also require some special attention, but we're able to get good EEG with all hair types, with the high density EEG. And with the headband, all of the electrodes are on the skin, not the hair and scalp. So there are no ethnic or racial limitations for that.
Okay, great. Last question. I know I already mentioned should have mentioned some of the ongoing study here. So I know this is relatively new. Everything is at the starting point. I think you guys did great. I mean, sounds very encouraging. But how can we help you? Or what kind of things potentially we can do together? And that is in my mind. I mean, of course, we can benefit. I can see that. But are you looking for some of the new collaboration or other things we can do?
We'd be very interested, particularly in your Latin America studies, because it sounds like your interests are very similar to ours.
Yes.
And we would definitely be interested in collaboration. Our limitation is funding. This is why we've set up crowdfunding. We've had fairly reasonable results from NIH, but we do not have NIH funding for our insomnia study. We just had a mediocre review from NINDS reviewers, which I think is pathetic because we clearly need this insomnia study and data. So our limitation will be funding. We can't provide you with any funding for those studies, and we would need you to have the funding to buy our systems. Now they're available for sale, and we can support them and help your work, but we can't provide any funding for it.
No. That's what I'm asking. I bet. I think that's why you have the NIA, SBIR grant, and this is one of the things you usually work with also the other companies before. So, well, I think
that's
we
right.
can keep that in mind.
Well, in fact, maybe we could collaborate on a grant project together. That might be a good point. If you have preliminary data, and we're scrambling to get preliminary data, and the SBIR might be a mechanism. We have to think about the innovation, et cetera, but we would be very interested in a collaboration with you.
Yeah, since you have the experience, because the program office always talk to me about that. Because the STTR, I always got the email. They asking people to apply. The funding rate is relatively high there, at least, I don't know, because the dropping maybe this year, overall funding dropping this year, but at least until like a few months ago, I always keep the email from the program officers told me that. But it's good that you have those things, and then we can think about more. Shahab, I think maybe we can think about what we can do, and then see for which project we can start using such a device to see if it's working or not. I think the feasibility is the major concern for me. That's the we do not want to increase the burden too much, but if it's feasible, yes, I definitely we can try. Any other questions for the device?
That's right.
Things for here?
Yeah, if we still have time, yeah. So I do have a question actually. I think this is a fantastic work. But I think that some of those scientific interpretations might be a bit too early. So I want to step back a little bit. So I want to ask something basic. The first one is basically the slow oscillation, the localization. So I think your focus is slow oscillation. Then why do you really need high density EEG? So I'm thinking, for high density, maybe, I mean, you want to localize the source of the slow oscillation. I may think that if you need to localize some high frequency component, then you need the high density things to localize those things. So I'm just trying to understand the logic here. So why do we need high density EEG to localize the slow wave?
Yeah, related the question is that possible the source is different for different people? You need to high density EEG to figure out where the source of the slow wave from?
That's a separate, let me break these questions up. So the first question is, why do we need high density for localization? And it's a simple physical problem. If you, the ambiguity or uncertainty of the source localization is reduced by having more data. So the high density gives us many perspectives, many geometric angles on the sources. If we only have, let's say, 10, 20 electrodes, then the uncertainty, the region of uncertainty for the slow oscillations is much bigger. So for example, we could tell they're coming from the middle of the brain. We could say slow oscillations are deep. They may be in temporal, deep temporal and frontal regions, but there's a lot of ambiguity. And the high density is cheap and easy to use. That's the main point. It's not very expensive and it's not very difficult to apply it. So why would I use an old-fashioned electrode system when I could easily put 280 channels on and then have the precision of localization? So it's a relative degree. You can try to localize from a low channel count 1020 system, but the uncertainty is much higher.
Yeah.
I would also argue that because this is the black source, identification, the old problem. People use many channels to to identify the blind source, right? I think this is an old system, but I can easily argue that for high density, there are a lot of things they are highly correlated. So that's why I'm thinking whether you really compare the localization problems without high density.
Yeah, there's a lot of literature on that. In fact, I'd be glad if you send me a note, we can follow up with localization literature. And so it's an asymptote. So if you try to localize from 3 channels, it's very difficult, not impossible. You can do some estimation. If you go up to 16 channels, the localization gets better by a lot, actually. As you go to higher densities, there's an asymptote. The uncertainty declines more slowly from 128, which is a fairly optimal level, to 256 or 280. But it declines at every point. The uncertainty of the source localization is asymptotic, but it continues to decline. And you have to understand those variables to address that.
Yeah, I think in general it makes sense. And I don't know if you have a measure for the size of the source. So for example, if that's the case, then if you have very high density EEG, and then you can minimize you can be more accurate in identifying the source.
That's correct. And it's really the region of uncertainty.
Right.
The same principles apply, but you have a large area of uncertainty with a low density. For example, 32 channels, you can do localization, but in a fairly crude region. As we go up to higher densities, 280, now we have a fairly tight localization, and the error of our estimation is much less. And this is easily shown in mathematical studies. So if you have an accurate head model, which is essential, you have to know all the conductivity of the head tissues, and that does become individual for a single person. Then you can do simple mathematical studies of putting a source at a certain region, generating the fields, and looking at the recovery. And we and other people have done this. There's a fairly well-established community of EEG and MEG source localization. And, I summarized the basic results. You decrease uncertainty, but with an asymptotic return. So you get the best results at 64 or 130 for the lowest channel counts, but you still get improvement up to 280. And the cost is not that much, particularly the cost per patient for a 280 system is really negligible. If you're doing more than 50 patients in a study, you're really wasting a lot of money and time by using low density EEG.
Yeah. Yeah, Dr. Tucker, I mean, thank you so much. I mean, because we have another event soon, so I want to keep a lot of interesting things we can discuss together. I mean, scientifically and also application. So maybe we can still keep contact by email. Maybe schedule another time to talk about the potential, collaboration or grant submission, whatever we like to do together. Okay. So Shihab, I think you are the key contact person here. So any other things, Dr. Tucker, you have a question for us before we leave?
I would say send me any papers or descriptions of your current projects. Maybe that'll help us get a creative dialogue on some possible collaborative funding.
Oh, definitely. Shihab and I are going to do that. Okay. Again, thank you so much for the wonderful presentation, and then we learned a lot, of course, trigger lots of questions. I silenced them now because we do need to move to another event. Okay. Thank you so much.
That's great. Great. Thanks
Thank you very much.